took our documents. Vito has several years already GLP. It's a laboratory who is well known for GLP, and our laboratory is on the track to get GLP. So we both, Vito and Nilu, we are responsible in Q Nano project for GLP tasks and training and hoping that also our round robin will be run in the spirit of GLP. And so Inge took actually our documents for study plan, for study report, and also we chose representative standard operating procedures which are related to this particular study. And just to explain this study was actually run in our laboratory as a training how study should be run, because we expect in May second pre-inspection where this will be inspected. So we want to kind of practice how we implemented our GLP system, not only on documents but also in laboratory. So these are my colleagues, Anya, who helped enormously with developing standard operating procedures, and also she will tell you how we actually deal with handling of nanomaterial and safety, because that's what we also learn during this conference. It's a huge variation with how laboratories and about concern and handling of nanomaterials in nanotoxicology laboratories or laboratories which deal with nanomaterial. And also Yugandhar, who is here, he is actually master student in quality assurance and GLP in our laboratory, and he is helping to our quality assurance to develop the system and to inspect system. So, and also he in this particular study, he was the inspector, so he will tell you finally what he found and what did he inspect. And so if you look at your documents, you have a study plan. Don't look at study report, because this exercise will have two parts. One, you will look at the study plan, and then look at what should be there, what is obligatory, and what probably is missing, or what is the critical. And this particular study plan, I don't know if I should repeat it, because Phil already said that what study plan should contain. It should have objectives and experimental design for the conduct of non-clinical health and environmental safety study. And adhering to GLP principle, the goal is to enable to full reconstruction of any study. So this study could be repeated with, of course, by trained personnel or trained person. And as it was already said, the important person is study director, QA, and also study personnel who are performing study. So each study should have identification, so you can look at it according to study plan. Study should be conducted, and all data should be recorded directly, promptly, accurately, legibly by the individual entry of the data. And any change of data shouldn't obscure the previous entry, and so all changes should be indicated and signed and dated. So it should be clear track what is going on, as we had the example with dead animals. So these kinds of things shouldn't happen. So these are the major points or major things of content of the study. Identification, descriptive title, nature and purpose, so it should be clear what will be studied, identification of test substance and the reference substance to be used, and also information concerning sponsor, test facilities. Test facility is laboratory. We also had to learn this terminology, study director, test facility, and this. But now it is quite clear in laboratory. So it should have dates, as it was said, and you can look at the study plan. It should have clear description of the method, reference to OECD guideline if there is any, and justification why we selected this test system, and characterization of test system. And also lots of information about, if available, about the test substance, identification and registration, safety precautions, preparation of test solutions, reference substances, identification, positive and negative control, and preparation of control solutions. So you will have a look to this study plan, and check if all is there, and also if the method and condition of exposures, doses are there, and how it is, if it's recording described, and that should have a study distribution plans, study plan distribution, quality assurance, information, and references. So we have now this we chose the simple study also for our training, just to be able to run it, not to run complicated study from start. So as example, it is a silica nanoparticles tested in vitro test method, or by in vitro test method, simple cytotoxicity test for plating efficiency. And we followed study plan. We followed plating efficiency standard operating procedure, and also you can see two additional kinds of materials, and one is actually form, study operating procedure form, where we actually have a template. When we run the study, we already have a form where we fill the date of all identification and all what was happening in that during that method, how many cells, how the calculation, pipette use, so everything is already pre-template, so you just write a number and number of cells. And so we try to make our life easy, because for us it seemed too complicated to have for standard operating procedures for everything, follow exactly the same procedure. So we want also us to take it in easy way, and we strongly recommend actually this form and templates, because it's also minimize the possible variation in reporting the data, and also simplify and make our life easier. So you could also see product technical sheet. And there is one mistake here, that study plan is for 25 nanometer size, and product technical sheet is for 50 nanometers, but we have correct thing in our laboratory. This I just described very briefly methods for you to be able to follow this study plan and standard operating procedure. So we simply see the cells then treat cells with chemicals or nanomaterials, and then after the treatment we trypsinize them and inoculate them in very small inoculum, like 100 cells or 200 cells, and after a certain period, each viable colony, each viable cell makes a colony. So simply just counting colonies, we we see what is the cytotoxicity. And maybe here it is even better illustration that you treat the cells and then inoculate the small, and when it's toxic, so then of course cells die, and if it's not toxic, so each viable cell can make a colony. And this is colony forming or plating efficiency assay, and we can calculate simply percentage number of colonies against cell inoculated times 100, so it's our percentage of plating or efficiency of plating, percentage of plating efficiency. And we can calculate IC50, so the concentration which induces 50% of death. So the question here will be, identify critical phases in the provided standard operating procedures and study plan that are related to this particular study. So I don't know how we can run this. We probably have half an hour. You can read and check everything, and we are here to help if you don't understand something.
Do you want to split them into groups, or do you want?
Yes, I was thinking about there are 30 of them, roughly. So 5 groups for 6.
5 of 6, yes, fine
Yeah.
with me, yeah.
So if you could make 5...
5 huddles of 6.
Maybe even to go with all your documents somewhere to sit for
Upstairs.
Upstairs for half an hour, because then we will have lunch, and after lunch we come and we can write, or you can make some points what is really critical phase. But think of it from the point of view of this particular method, this particular study, but also from the point of view of nanomaterial or nanotoxicology. So what is really critical for this?
One thing I was going to say that has been my experience when doing workshops like this is that people from the same organisation tend to sit together.
Don't do that.
And I don't want that, so what I'm going to do is I'm going to split you up. That way you get to talk to somebody else and share their experiences, which is half the fun. Now, you say there are 30 people in this room, yeah?
Yeah, roughly.
So what I'm going to do is I'm going to give you that back, and I'm going to number everybody. This is your group number. So you go into whatever group. So you're 1.